Hello everyone, this is Dr. Pritpal Singh, an MBBS MD from CMC Ludhiana, and today I'll be discussing about the competency PH 1.4, which says describe the uh, absorption, distribution, metabolism, excretion of drugs. Today we will be focusing only on the various terms that are used in pharmacokinetics, and we would also be talking about certain other terms that are there. And these terms would be for our focus only would be on absorption today, and then proceedings this we'd have few more sessions on absorption, distribution, metabolism, and excretion. So we start with the term pharmacokinetics. Now, this term pharmacokinetics is a quantitative study uh, of drug, and it said it is a study of drug movement in, through, and out of the body. Or let's say in simple term, it says what the body does to the drug. This is the basic definition, or the other form that is also used is absorption, distribution, metabolism, and excretion of the drug encompassed in the term pharmacokinetics. So why do we require to study pharmacokinetics? But obviously pharmacokinetic is an important parameter or an important aspect that has to be studied because we need to know how a drug gets inside the body, what happens when this particular drug is inside the body, and how does the body get rid of the drug. So that is what is said in pharmacokinetics, that it is the term that says that what the body does to the drug. Now, moreover, the reason why pharmacokinetic is required because it helps us determine the route by which the drug is taken, the dose in which it is to be taken, the latency of onset, that means the time it would require to start having its action, time to peak action, and the duration of action, obviously, which is important to know the frequency of administration of drug. So the major organs involved in our body, let's say for giving a drug by the oral route, are gastrointestinal route where absorption takes place. This is followed by the drug enters the liver where metabolism takes place and it is excreted by the kidney. Although lung also serves as one of the methods by which absorption and excretion of the drugs takes place, this too specifically for volatile anesthetic agents. So we first start with one of the components that is one of the criteria of pharmacokinetics, or one of the important parameters for pharmacokinetics, that is absorption. So in simple terms, absorption is movement of a drug from the site of administration to the circulation. That means once it reaches the circulation, absorption is over. Now this absorption would depend upon, obviously it is not important, or we don't need to take this into consideration when we are talking about the IV route, because by the IV route we are directly injecting the drug into the circulation. But when we give drug, most importantly by the oral route, there are many factors that come into play, that the first of them being the aqueous solubility of the drug. The more the water soluble the drug, the lesser it reaches the systemic circulation, the lesser of it is absorbed. Uh, other point is it would depend upon the area of absorbing surface. By default, ileum or the small intestine is the part that where drug is maximally absorbed. Depending upon the drug concentration, as I said, movement from a higher concentration to lower concentration, the vascularity of the absorbing surface, higher blood supply favors drug absorption, lower blood supply is somewhat ample. Particle size, smaller particles tend to get in easily, larger particles require some particular special mechanisms to enter. The route of administration, whether we are giving the drug by the intramuscular route, by the subcutaneous route, or by the oral route. Then with food, there are certain drugs or certain components in the food that tend to form complexes with the food, such as calcium in the milk that forms a complex with tetracycline. Other important aspect is the GI motility. If there is a faster gastric emptying, that means the stomach, we hasten the gastric emptying of the stomach, it accelerates the drug absorption. Ionized or pH, acidic drugs or basic drugs, they require specific media to be absorbed. And obviously there are certain drugs that get degraded in the GI, example penicillin, which is degraded by the acidic environment in the stomach, and insulin that is degraded by the peptidase, because being a 51 amino acid, it gets degraded. So for absorption to take place, we need to remember this particular membrane that is there. This is the plasma membrane, which is a bilayer membrane. We have the hydrophobic fatty acid chain that is there, the hydrophilic polar heads, and then we have the transporter proteins. Some proteins are transversing through the entire membrane. Some are only on the outer membrane, some are only on the inner membrane, and all of them do have a specific function. Now, this is one of the major barriers that tend to hamper or favor drug absorption or drug movement. And the first we need to discuss the main mechanisms of transport across the biological membrane. So this could be passive diffusion, facilitated diffusion, active transport, endocytosis, and filtration. Facilitated diffusion and active transport require a carrier transport for their action, that we were talking about certain proteins located or traversing the membrane located at the outer aspect of the cell membrane or on the inner aspect of the cell membrane. So let's talk about first phenomena for drug movement across membrane, that is passive diffusion. Passive diffusion simply means movement of the drug from a higher concentration to a lower concentration. So this is the lumen, and because in the lumen we have maximal concentration of drug and it there's lesser amount of drug out here, so there will be movement of drug, and this is just a movement of drug from higher concentration to lower concentration, and this by far is the major mechanism by which movement of drugs, all drugs, occurs inside our body. So this is the most important mechanism, and it applies to non-polar drug, that means a lipid soluble drug. The concentration gradient, I've already been telling you about it, is a driving force, and we do not require energy for it. The next one is the facilitated diffusion, where we have a carrier transport. Now this carrier transport, it could transport the drug inside the cell and maybe something out of the cell. Then it would be an antiport, or it could drive the drug as well as something inside the cell. Then it would be a symport. And similarly, again out here, the driving force is the concentration. We have the higher concentration in the lumen, lower concentration inside the cell. So there is movement of drug inside the cell, and so on. So the major difference between passive diffusion and facilitated diffusion is the carrier transport protein that we are talking about. There are fewer drugs that can be transported upon with this particular mechanism. It will depend upon the carrier protein. Obviously, concentration gradient is required and there is no energy required. One of the example is diffusion of tetracycle inside the bacteria. Then we talk of active transport, where we require a carrier protein as well as energy. Now this energy is in the form of ATP, and this ATP is converted to ADP. So the drug, it moves from a higher concentration or maybe a lower concentration to a higher concentration with the help of energy and carrier transport. So this particular thing can proceed against the concentration gradient as compared to passive diffusion and facilitated diffusion, where the movement is from higher concentration to lower concentration. The other phenomena is endocytosis, which implies to drugs which have a very high molecular weight. They cannot be transported as such, so they are engulfed in the inside the cell, and this particular phenomena is endocytosis. So there is internalization of the large molecules of cells which have a molecular weight of more than 100, examples being hormones and immunoglobulins. The fifth process by which drugs can cross the cell membrane are when they pass through the aqueous pores in membrane or via parallel spaces, such as blood capillaries and glomerular capillaries. The other important factor that determines the absorption of the drug is the pH, which is the negative logarithm of hydrogen ions. And most of these drugs are weak electrolytes. So that means their ionization is pNH dependent, and ionized drugs being water soluble, aqueous solubility, they do not cross the cell membrane, whereas unionized drugs can easily cross the cell membrane. So how this particular thing works? So let's say for a weak acid HA, this is dissociated into H positive ion and A negative ion, and the pH is determined by the dissociation constant and log concentration of the negative ion over the acid. So the pK is a negative log of dissociation constant of weak electrolyte. So weakly acidic drugs ionize more at alkaline pH and they are not absorbed, whereas weakly basic drugs ionize more at acidic pH. So this particular thing leads to a phenomena that is known as ion trapping. Now, what is the significance of this ion trapping? Let's talk about this. So as by virtue of what I have been telling you, acidic drugs remain unionized in the acidic state and basic drug remains unionized in the basic state. That means that acidic drugs should be absorbed in the stomach and basic drugs should be absorbed in the intestine. So let's talk about an acidic drug, aspirin. Now what happens is this acidic drug, aspirin, it being a stomach being an acidic media, it gets it is unionized and it enters the mucosal cell. Now inside the mucosal cell, the pH is alkali. So this particular drug gets ionized out here. And because of this ionization, uh, this ionized molecule is not able to get passed through the cell and gets trapped inside the cell, which is known as ion trapping. Now this ionized molecule is very slowly converted to the unionized form and it slowly passes to the extracellular fluid. So this could be one reason why gastric mucosal damage occurs with aspirin, because aspirin being an ionized drug, it would enter the mucosal cell. Mucosal cell has an alkaline pH where it would get ionized, and ionized molecules being water soluble gets trapped inside the cell, where they are slowly released into the extracellular spaces. And this is one reason that I said that gastric mucosal damage occurs with aspirin. Similarly, basic drugs have higher intercellular concentration when the pH ranges from 7 to 7.4. Now we come to another term that is there, that is bioavailability. Bioavailability is the fraction represented by F. That is the fraction of a given dose that reaches the systemic circulation in the unchanged form. And then when we talk about factors responsible for absorption, we excluded the intravenous route because in intravenous route, the entire drug reaches the systemic circulations. Hence, bioavailability by the systemic circulation is 100%. By the IV route is 100%. Now, this fraction of the drug that reaches the systemic circulation, it tends to influence the rate and extent of absorption of a drug from a particular dosage form. So IV has 100% bioavailability, subcutaneous or intramuscular route have almost 100%, whereas by the oral route, there is always less than 100% bioavailability. So this is due to incomplete absorption, the first pass metabolism, and most of the drugs are excreted in the bile. So this bioavailability would depend upon area under curve, the maximum concentration of the drug, and the maximum time for which the drug stays. There are certain variations in the bioavailability such as disintegration and dissolution. Disintegration is breaking down of larger molecules into the smaller molecules that do tend to play a role. Now, this bioavailability is important for drugs which have a low safety margin such as lithium, and where you require precise dose regulation such as giving oral hypoglycemic drugs. So this is a graph which represents giving a drug by the IV route, the oral route, and the rectal route. You will find the area under curve is maximal in the IV route, and hence bioavailability is maximum as compared to oral route and lesser as compared with the rectal route. Now we are talking about disintegration and dissolution that are responsible for bioavailability. You give a tablet, this tablet is broken down into smaller molecules, that is disintegration, and then this smaller molecules are dissolved in the gut fluid, that is dissolution. Now this dissolution leads to absorption, where it reaches the portal or portal blood, it reaches the liver, where a variable amount is metabolized and the remainder reaches the tissue that is there for action. So that is what bioavailability is given by the oral route. So factors that determine bioavailability are disintegration, dissolution, and the liver metabolism or the first pass metabolism that is there. So this is all regarding the absorption, and I hope I have been able to put some light on absorption. And if there are any queries as such that you feel are there, you can get in touch with me, or you can write on the chat box and I'll definitely get back to you. Thank you so much for your patient hearing and happy learning.